researches are in the field of cancer biology, medicinal plants, and clinical biochemistry. He guided 10 MPhil researchers and 4 PhD researchers. 52 research papers were published by him. He had participated in 25 conferences and organized 4 conferences. He had completed UGC minor projects and TNSCH mini projects. He is the reviewer in Indian Journal of Experimental Biology and chairman in Board of Studies. He had received a Proficiency Award and Best Paper Presentations Award and also participated as coordinator in Red Ribbon Club and Rotaract Club. Gladly, we welcome you, sir.
Okay. Sir, shall I start my lecture?
Yes, sir.
Yes, okay.
We can start. Welcome you, sir. We are hoping for a great lecture from you.
Okay.
And I have seen your CV, and it was very exhaustive CV, sir. And with your experience, I hope that our students will get excellent knowledge from you, sir.
Definitely, sir. Respected correspondent of this Paavai Engineering College and secretary and the principal of your reputed college and the head of the Department of Pharmaceutical Engineering and Technology and Biotechnology and faculty of your college and my dear students from Pharmaceutical Chemistry, Biotechnology and Biomedical Engineering. A very warm good evening to one and all. I am very happy to deliver my lecture on enzymes in industry and diagnosis. Next slide, please. Next slide. Karthi, okay. First, let me introduce what is enzymes, right? Actually, you know that in plus 1 and plus 2, we learned enzymes, right? Basics of enzymes. So enzymes are protein in nature. All the enzymes are protein in nature except ribozyme. So you know that this ribozymes is like RNA acting as enzyme, right? And moreover, these enzymes are organic biocatalysts. You know what is organic? So organic means the compound which is having carbon in its structure, right? And these enzymes are acting as biocatalyst. That is, these enzymes are speeding up the actions. And moreover, all the enzymes are functional units of metabolism. That is metabolism, you know, that is it is made up of two components, that is anabolism and catabolism. And moreover, all the enzymes are specific in action and they are very accurate in their action. And they have active site in its structure. All the enzymes, they have active sites. That is, active site means it's a part of enzyme to which substrate can bind to form products, right? And these enzymes are colloidal in nature. All these things are colloids, right? It is colloids means semi-solid in nature. It is not very liquid. It is not very solid. And moreover, all the enzymes are influenced by temperature and pH and substrate. Like us, we are all influenced by temperature and pH, right? So likewise, the enzymes are also influenced by temperature, pH, and substrate, right? Next slide, please. And what is the purpose of using enzymes in food industry and also in some industry? There are reasons, right? And there is some of the reasons are to modify the food and also to improve the nutritional quality of the food items and also to extract the enzymes, right? To extract some of the compounds, these enzymes can be used, and also these enzymes are used in brewing industry. And this brewing industry is like it is wine industry, beer industry, and some other distillation product process, right? And also for the fragmentation of these compounds to enhance the color, texture, and also appearance, and also taste, right? So for this purpose only, enzymes are used in industry, that is especially in food industry. Next slide. And what is the need and the significance of the enzymes in food industry? As I told you before, that is to enhance the quality of food, that is high quality products, and moreover low cost of the production of the materials or food items, and low wastage, and also to minimize the energy consumption during the processing, that is food processing industry. Next. And you may ask, what are the sources of the enzymes, right? So from where do we get? From where do we get? Dear students, please.
Please mute your mic now.
Please mute
Yeah,
your,
please.
mute your audio, please.
Yeah, it's disturbing me. Yeah, please.
Okay, thank you very much. Right, dear participants, kindly mute your audio, please. Okay, and from where do we get these enzymes? Right, what are the sources for the enzymes which is used in food industry? That is, there are three main sources. It is plants, animals, and microorganisms. Right, and from plants we get these enzymes. There is alpha amylase, beta amylase, and beta glucanase, and bromelain. It's one of the excellent enzymes. That is, it is like a protease, and papain, that is from papaya, and chymopapain, and also lipoxygenase. Right. So these are all the enzymes which is obtained from plants. And coming to the enzyme which are obtained from animals, it is trypsin, chymotrypsin, pepsin, renin, catalase, amylase, and pancreatic lipase. Right. So coming to the microorganisms, so microorganisms excellent source for enzymes like glucose isomerase, alpha amylase, and beta amylase, right? So these are all the sources for enzyme which is used in food industry. Next slide, please. Okay, and there are mainly four types of industries in which we are using the enzymes. There is first one is dairy industry, and brewing industry, and baking industry, and wine industry, and fruit juice industry. So the table, that is what are the enzymes used in this industry, is given here, like rennet, lactase, protease, catalase in dairy industry, and brewing industry. There is glucanase, amylase, protease, and amyloglucosidase. These are enzymes used in brewing industry, and enzyme used in baking industry is malto amylase, glucose oxidase, and pentosinase enzymes. And wine industry, pectinase, and beta glucanase. These are all the enzymes used in different types of industries. In that is in food industry. Next slide, please. Especially in dairy industry, this rennet, lactase, and protease, and catalase, these enzymes are used in a batch-wise process. That is rennet. Actually, another name is renin, used in dairy industry to produce or to manufacture the cheese. And lactase, it is also used in milk industry, and protease to cleave the protein, and catalase, it is the best enzyme. It is for the processing of food. Next slide. Yes, okay. Thank you very much. Please, that is, turn the slides as early as possible. Rennet, coming to the rennet, another name for this rennet enzyme. Actually, this enzyme source is obtained from the young calves. Actually, that is now the obtaining of this renin or rennet from the calves is actually it is banned because we have it is a committee, that is animal cruelty committee is there. So that committee now banned the extraction of enzyme from the animals, right? So instead of that, we get the enzymes, that is rennet or renin from the microbes, right? And these enzymes cause milk to become cheese. So this rennet is mainly used in cheese making industry, and also separates the solid curd and also liquid whey. You know what is whey? So this whey is nothing but it is upper layer of the curd, that is called whey. So whey is rich in sugar, that is especially glucose and also galactose and lactose, right? So best source for glucose, this whey, and this whey is also used in candy industry, right? And these different types of cheese we can get, but one of the famous cheese, right, variety is available. That is thistle. Actually, the thistle is a type of cheese which is having a good quality, right? From the which is produced from the milk by using the rennet. Right. Next slide, please. Please make it fast. And coming to the lactase, so this lactase, this enzyme which acts on lactose. You know what is lactose? Lactose is a disaccharide. Actually, it is present in milk, right? And this enzyme lactase, it is also extracted from the yeast, that is lactase enzyme. And these enzymes, it is mainly used for digestion of the whole milk in order to avoid indigestion, right? And this lactase is also used in mainly for two industries, that is in the ice cream industry and also in candy industry. This lactase, that is to produce or to manufacture the ice cream, it should be very soft, right? So for soft candy and also to make this soft ice cream, we need this lactase, because this lactase removes lactose which is present in milk, and also it digests the lactose which is present in milk and make the ice cream soft, right? So this is the main use of lactase in ice cream industry. Next slide, please. Okay, this catalase, another excellent enzyme. Actually, it is produced from bovine liver, also from microbial sources. What is the function of this catalase? This catalase actually breaks down hydrogen peroxide, that is H2O2, to form water and molecular oxygen. You know that this hydrogen peroxide is very toxic in our body and also in food items, right? So in order to in order to remove this hydrogen peroxide in food items, we are using this catalase, right? So along with the catalase, we can use glucose oxidase to prevent oxidation in the food wrappers. So when you wrap the food items, it forms oxidation, right? So in order to prevent oxidation, both catalase and glucose oxidase can be used in food industry, that is mainly for packaging of food. Okay, and it also removes the hydrogen peroxide. That is small amount of even small amount of hydrogen peroxide can be removed by using these enzymes, right? Next slide, please. Coming to protease, actually this protease is widely used enzyme among all other enzymes. As the name implies, protease, it acts on proteins, right? It acts on peptide bond and breaks the protein to form amino acids, right? So another excellent enzyme used in food industry. So it hydrolyzes the peptide bond and also it forms the cheese, right? It is used in cheese manufacturing, and also it results in bitter flavor to cheese. This protease not only breaks down the protein molecule, but also it add the flavor, texture, and also flavor to the cheese. Right? Next slide, please. Yes. Please make it fast. Okay, and coming to dairy industry, it is used in many other food items like milk, ice cream, yogurt, and cheese. That is, what is the purpose of using these enzymes in this dairy industry? That is to convert the sugar into digestible form of sugar, that is disaccharide into monosaccharide, and also to form simple sugars like glucose and galactose. So in order to make the food items in a complete and a nice texture form, and also in order to improve the flavor, right? So these enzymes are used in milk, ice cream, yogurt, and cheese items. Next slide, please. In cheese manufacturing, there is apart from rennet, we can use lipase also, right, to impart flavor. And in candy making, invertase and lactase are used in candy. It is for sweet making, and to produce the white colored bread, right? So bread whitening, right? Bread whitening. So it can be obtained by using lipoxygenase enzyme. And the next is clarifying fruit juice. That is, what is clarifying fruit juice means to get the fruit juice in a clear format. That is transparent juice without any debris, right? So clear juice by using this pectic enzyme, because the fruit outer layer, it has pectic substances, right? So in order to cleave the pectic substances, we can use this pectic enzyme. So when we use this pectic enzymes, it cleaves the pectic layer, outer layer of the fruit, and make it a clear juice, right? So for clarifying fruit juice, we can use pectic enzyme. And next is desicing fabrics, right? So another important uses of enzyme, that is, for this we can use amylase enzyme. So this amylase, what does it do? Actually, this amylase, it acts on starch to form glucose molecules. So for desicing fabrics, there is in textile industry we are using amylase. And next one is tenderizing meat. Actually, what is tenderizing meat? Meat means tender means to make it soft, right? So to form soft nature of the meat, we can use this protease, right? That is mainly in chicken and mutton. We can use this protease enzyme to make the meat very soft nature, right? So protease, mainly two types of protease we can use. One is bromelain, and another one is papain. So bromelain is actually it is obtained from pineapple, and the bromelain it is obtained from actually papaya, right? So these two fruits are very important to deliver the protease, right? Excellent source for protease, which is used in tenderizing the meat. Next slide. Okay, and next slide. Next slide. Next one. Okay, and in brewing industry, these are the enzymes used: protease, beta glucanase, alpha amylase, and amyloglucosidase. These are the enzymes used. You know that the function of the enzyme, it is protease used to break proteins. Glucanase, it is used for digesting the dextrin and the dextran compounds. Alpha amylase used to break starch, to convert the complex sugars into simple sugars, right? And amyloglucosidase, it also acts on amylopectin, that is a compound of starch to make it simple sugars, right? So these are all the four types of enzymes used in distillation and also distillery industry, and also in the brewing industry. Next slide. Yes. Next slide, please. Next slide. Next slide. Next slide. Okay. These protease acts on larger protein molecules, and also it reduces the haze in beer item, right? So beer and wine item, dear
Hello,
participants,
please mute your
mute your mic, please. Mute your audio, please. Hello? 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 Please move it fast. Next slide.
Listening, sir.
Next slide. Move it to next slide. Okay, please make it fast. And so this beta glucanase, actually this enzyme acts on a glycosidic bond. You know that one glucose bind to another glucose by means of glycosidic bond. So this beta glucanase acts on these bonds, right? So what is the function of beta glucanase? It is also used in massing and also in brewing industry. It is massing means to mix the complete texture or complete compound, complex compound into simpler one. That is massing, right? Next slide. Next slide. Alpha aminase, you know that it acts on starch, is also used in massing and also in brewing industry. Next slide. Next one. Next one. Next. Amylate glucosidase used in that is helps in improving taste and flavor. Next slide. This is distillation industry process or steps involved in brewing industry. Next slide. Next one. Baking industry. What is the purpose of the enzymes used in baking industry? That is in bakery products. That is in order to improve the appearance and also to improve the freshness of bakery items and also to improve taste, flavor, and also color. So that is for this 5 important reason we are adding the enzymes, right? That is appearance, freshness, and also taste, right, and flavor. And in that bakery industry, alpha amylases are used to produce dextrin. Dextrin is nothing but, I say, actually, it's a carbohydrate compound which is broken down to sugars. And beta amylase, it also used in yeast fermentation and also increases the volume of bread. And proteases, it also used in baking industry to improve the elasticity in biscuits and also in wafer manufacturing, right? And hemicellulose, another type of enzyme, it improves the duff properties and also bread quality of the food items. And lipoxygenase, already I told you that is used in for whitening the bread. That is for bread whitening process. Next slide. Next one. In baking industry, three more enzymes are used. Maltogenic amylase, glucose oxidase, and pentosinase enzymes are used to improve the texture of bread. Next slide. Next one. Maltogenic amylase, it acts on maltose. And you know that maltose, actually, it's a monosaccharide. It is this amylases, it has anti-staling property. So what is anti-staling properties means it prevents spoilage of food items, right? This enzyme, it is mainly it is used to used in food items to improve the period, that is life period of the food products, right? And moreover, it modifies the starch, to improve the texture of that is duff and other food items. Okay, next slide, please. Glucose oxidase, it is like catalase. This enzyme, it acts on hydrogen peroxide, and this glucose oxidase actually it removes the H2O2 and also prevent oxidation in many food industries, mainly especially in the bakery industry. Next slide. Next one. Pentosinases, another beautiful enzyme, right? So it also improves the duff quality, and moreover, it is easier to handle. After using this enzyme, it is very easier to handle the duff, okay, which is used in the bakery industries. And moreover, the duff is more stable after using this enzyme, that is pentosinase enzyme. and also it gives a better oven, right? Oven spring during this process, right? And this wine and fruit juice industry, these two enzymes are used. This pectinase and beta glucose enzyme is used. Next slide. Next slide. Next slide. There is fruit juice industry. Adispectic enzymes are used. Pectinase enzymes are used to improve the juice volume and also for juice easier method of juice formation or juice production. Next slide. Next one. Pectinase already discussed. Next one. There are so many applications of pectinase, like it is used in textile industry and degumming of fibers, wastewater treatment, paper making industry, and coffee and tea fermentation, oil extraction and brewing industry, bioethanol production, and also used in paper industry, right? There are many applications of pectinases. So it is a wide spectrum enzyme used in most of the industries, right? And the meat industry, to make the meat very soft, that is tenderizing of meat, these enzymes are used. These enzymes soften the muscles and also make the meat very soft, right? And one more enzyme is used in this meat industry, that is trans glutaminase, right? So this trans glutaminase, it is used to induce gelation or gelatination in this meat product, right? That is, it removes most of the proteins in this meat industry. And coming to coffee and tea industry, it is coffee industry enzymes are used to liquefy the mucilage of coffee cherry, right? So to make it very good coffee. And some of the enzymes used in coffee industry are cellulase, which acts on cellulose, and hemicellulase acts on hemicellulose, and the pectinase acts on pectic compounds. And coming to tea industry, some of the enzymes are used, like cellulase, glucanase, pectinase. These are all the enzymes used to break down the cell wall of tea leaf, right? So tea leaf.
Hello, I think we lost you, sir. Hello, Jayaprakash, sir. We are not able to hear you.
Yes. Now it's audible, sir. Okay.
Yes, sir. Now, yeah.
Okay. So in the coffee and tea industry, some of the enzymes are used, right, to improve the taste and quality of tea and coffee. In coffee, some of the enzymes like cellulase, which acts on cellulose, hemicellulose, and pectinase acts on pectin compounds are used. And what are the enzymes used in tea industry? It is cellulase, glucanase, and next slide, please. And someone is mutating my audio, right? And I don't know who is operating. And during the slide presentation, right, my audio is mutated. Dear organizer.
Okay. Let me check.
Okay. Dear organizer, don't mute my audio, right? So coming to detergent industry, right? So what is detergent and what is soap? My dear participants, what is the difference between detergent and soap? Yes? Is there any difference between detergent and soap? Or both are same? Hello? Actually, this detergent means which can able to act on there is hard water, right? Detergent, and the soap it is acting on it is soft water, right? Hard water, it has more amount of salt. Even in salt containing water, this detergent it do its function, right? It does its functions in detergent industry. That is difference. Moreover, this detergent used to remove the stains, right? And soap, it cannot remove the stains, right? So this is a small difference between detergent and soap. That is, these two things are different. Okay. So in detergent industry, in order to remove the stains, right, protease are used to remove stains due to glue or some vegetable sauce, right? So you know that vegetable sauce and glue, it is very hard to remove. So in order to remove this stain, which is due to glue or protein stains, this enzymes, protease can be used. So when we use this protease, it acts on proteins. So this protein is converted to amino acid. So once it is converted to amino acids, these amino acids can be easily removed by simple washing, right? So this is the action of enzyme used in detergent industry. So enzyme used here is protease, right? And also it is used in laundry industry and also in automatic dishwashing industry. We are using this enzyme protease. Next slide, please. Next one. Yes. Okay. So that's all about uses of enzyme in food industry. And coming to diagnostic applications of enzyme, that is enzymes used as a diagnostic system, right? So coming to the introduction, you know that enzymes like microbes, each and everywhere in human being and other industry process, enzymes are used in various types of industries, right? In human being also, this and other animals, there are nearly about 300 types of enzymes present. Okay, so to catalyze more than 1,000 types of reactions in our body, right? So the enzymes actually it is used for diagnosis of many diseases, right? And there are, as far as this diagnostic system is concerned, we can divide the enzyme into two. That is, one is plasma functional enzyme, and another one is non-plasma functional enzymes. Next slide.
Hello, sir. No audio, sir.
Again mutated, and I don't know, the organizers are mutating. And who is operating the slides? I don't know, but it's somehow it is mutated. Okay. So this plasma functional enzyme. So this there are two compounds which can be produced from blood. One is serum, and another one is plasma. So these things are very useful for biomedical students and also pharmaceutical engineering students. Okay. So when we add this anticoagulant, we get plasma. So without anticoagulant, after collecting the blood, right? After centrifugation, we get the liquid that is called the serum, right? So serum is without anticoagulant, we are getting the liquid that is called serum, and plasma is with anticoagulant. We get the liquid from the blood that is called plasma, right? Now I think you understand the plasma and serum, right? So these enzymes are released into plasma, right? It is when the liver is damaged, these enzymes are released, right? So example for these enzymes, there is plasma enzymes are thrombin, lipase, and plasmin and ceruloplasmin. These are all the enzyme, right? This is plasma functional enzyme. Next slide. Next one. Okay. Impairment of liver. Actually, when the liver is damaged in our body, right? So it leads to decrease in the activity of this enzyme. Okay. Level of enzymes in blood, it is decreasing, right? So when the liver is damaged, right? So example is deficiency of ceruloplasmin in Wilson disease, right? So what is Wilson disease is that actually when the liver is damaged, this level of the ceruloplasmin enzyme is low. Please go back the previous slide, previous one. Hello. Yes. So when the liver is damaged, the enzyme ceruloplasmin level is decreased. So this causes Wilson disease, right? So because of that deficiency, it causes a disease called Wilson disease. So this is also affecting the brain also, right? So it leads to memory loss, right? So this is Wilson disease. Okay, next slide. Next one. Dear participants, very few slides are there. Okay, next, non-functional plasma enzymes, right? So what is non-functional plasma enzyme means? These enzymes are not released into plasma, right? So this is the meaning of non-functional plasma enzyme. It is present in very low concentration in plasma, and
technical issue in what term?
Actually, decreased level of this non-plasma enzyme in plasma indicates the tissue damage. When our organ is damaged, when the tissue is damaged, the non-functional plasma enzymes level it may be increased. Okay, next slide. Next one. Yes. And what are the sources of this plasma enzyme? First one is cell damage. If any organ is damaged, the level of these enzymes will be increased. And also obstruction. What is obstruction? Obstruction means block. If there is any block in bile duct or bile in our body, it also increases the enzymes like alkaline phosphatase enzyme, right? So increase in the level of alkaline phosphatase it indicates the jaundice, right? Jaundice and also bone disease, right? Okay, next slide. Next one. Please make it fast. Yes. Okay. This when this non-functional enzymes it is present in higher concentration, it indicates damage. So a best indicator of damage, cellular damage, tissue damage, or organ damage. So this non-functional plasma enzymes level, it will be more. It is in the blood, right? So the increase in the level of this plasma non-functional plasma enzymes like protease and other enzymes indicates the cellular damage and also organ damage. Next slide. So what are the medical importance of these enzymes, right? These non-functional plasma enzymes, right? And mainly it is used in diagnosis of diseases, right? So what is diagnosis means? What are the process tests carried out during the screening of disease? There is nothing but diagnosis. What is prognosis? That is prognosis means treatment, right? Treatment after screening. That is called as prognosis. Okay, that is follow-up of this treatment by measuring the plasma enzyme before and after, right? So diagnosis means it's a testing method, and prognosis means treatment. Okay, so diagnosis and prognosis, and also this enzymes used in used as analytical reagents, and also enzymes are used as therapeutic agents. That is for the treatment purpose. Next slide. Yes. Next one. And coming to the liver enzyme, that is in the human being, you know that liver is the main organ for many metabolic pathways. Liver is the main organ that is involved in mainly many metabolic reactions, both anabolism and catabolisms, right? So when the liver is damaged, this enzymes level, it will be increased, like aspartate aminotransferase, alanine aminotransferase, gamma glutamyl transferase, alkaline phosphatase, and 5-nucleotidase. So when these enzymes level, it will be increased. When these enzymes are increased, it indicates the liver damage. Coming to aspartate aminotransferase, this is otherwise known as AST, AGOT, and GOT. So what is AGOT means? Serum glutamate oxaloacetate transaminase. AGOT is serum glutamate oxaloacetate transaminase, right? So this enzyme, this present in it is liver and also in muscle, and small amount of this enzyme present in kidney and pancreas. So when these organs, liver, muscle, kidney, and pancreas, when these organs are damaged, the enzyme level aspartate transaminase it will be increased, right? So the best indication for the liver damage, kidney damage. Okay, next slide. Next slide. So normal level of this AGOT is 14 to 20 units per liter. Okay, this is in male, and in female, this level is 10 to 36 units per liter. Okay, and what is the use of this enzyme in the laboratory? That is, it indicates viral hepatitis, it indicates cirrhosis, and also muscular dystrophy. That means muscle damage, and MI. So MI is that is myocardial infarction. So there is heart attack. Okay, or heart failure. Heart failure. So to indicate this diseases, this enzyme AST and AGOT is increased. Okay. And coming to alanine aminotransferase, it is otherwise known as AGPT. What is AGPT? Is serum glutamate pyruvate transaminase. I repeat this: serum glutamate pyruvate transaminase. So source of this enzyme is all tissues, including kidney, liver, and also muscle and heart. Okay. Next slide. This ALT. This ALT in male, the normal level is 10 to 40 units per liter, and female is 7 to 35 units per liter. So, being the biomedical students and pharmaceutical engineering students, you should know all these things when you go to the hospital, and also for testing various diseases in various organ, right? In any type of diseases, you must know what are the enzymes. It is diagnosed in the laboratory. Okay. So this enzymes level is, if it is increased, it indicates the hepatic disorder. So what is hepatic disorder means? Liver disorder. That is inflammation or any damage in the hepatic cells. That is liver cells. Okay, next slide. Next one. Yes. Gamma glutamyl transaminase, another enzyme, right? It catalyzes the transfer of amino acids from one peptide to another amino acid on another peptide. So this is function of gamma glutamyl transferase. Next slide. Next one. So what are the sources of this gamma glutamyl transferase? It is mainly present in these organs like kidney, brain, prostate gland in male, pancreas, and liver. So what is the importance of this gamma glutamyl transferase? It is mainly it is used to section or any block is there in the bile duct, and also it is mainly used in patients and also during pregnancy for bone disorder, and also in bone disorder this ALP level, alkaline phosphatase level, it may be increased, right? So to indicate the bone disorder and also for the liver disorder, this is used. And coming to this alkaline phosphatase, which is otherwise known as ALP. ALP means alkaline phosphatase, right? So this enzymes it is present in bone, liver, intestine, spleen, and also placenta and kidney. Okay, next slide, please. Next one. right? So normal level is 25 to 125 units per liter. Next slide. Yes. Lipase. Okay. So what is the function of lipase? It catalyzes the hydrolysis of triglycerides to form glycerol and fatty acid, right? So what is the source of this enzyme? That is pancreas, and mainly these enzymes used in laboratory to find out the pancreatic damage. That is within 5 to 8 hours, the lipase activity, lipase level, it will be increased, right? So this is the main symptoms in this pancreatic disorder. And coming to muscle enzyme, three types of enzyme: creatine kinase, lactate dehydrogenase, and glycogen phosphorylase. These enzymes, it will be more useful to find out the disease in muscle, right? Coming to the creatine kinase, otherwise known as CK or CPK. What is CPK? Means the creatine phosphokinase, right? So this enzyme catalyzes the reversible reaction, that is to form creatine phosphate and to from creatine, or creatine phosphate to creatine. And this reaction is reversible, both forward and backward, right? Next slide. Next one. So the CK, there are three types of CK: CK1, CK2, and CK3. Mainly the CK1 is increased in CNS disease. That is, CNS means it is central nervous system disorder, okay, in the brain. And CK2, the second enzyme, it is creatine kinase. It is elevated in myocardial infarction, that is heart disease and heart attack, right? And CK3, it is elevated in muscle disorder. Okay, so there are three types of CK. And as I told, it is increased in muscle damage and also in heart diseases, mainly in heart attack. The level is increased. Normal level of this enzyme is 22 to 196 units per liter. Next slide. Next one. Yes. Coming to lactate dehydrogenase, it is LDH. And dear participants, you should know all these things when you go for screening for any diseases, the hospital or in the laboratory, right? So this is the basic need, right? In order to maintain our health and hygiene. Okay. So this lactate dehydrogenase is LD or LDH. It catalyzes the lactate to pyruvate. So previous one. Hello. Previous slide. So this enzyme converts pyruvate to lactate and lactate to pyruvate. So this is also the reversible reaction. Next one. Next slide. So this LDH level, it is increased in mainly heart attack. It is heart disease, kidney disease. There are 5 types of LDH, lactate dehydrogenase, LDH 1, 2, 3, 4, 5. First one is increased in heart attack, and second one is kidney disease, and third one is increased in leukemia, that is blood cancer, and also malignancy, different types of cancer. And LDH 4 is increased in lung disorders, and LDH 5 is increased in muscle disorder. Okay, these are all the use of LDH in various type of diseases. Okay, next slide. Next one. Normal level of this enzyme is 100 to 200 units per liter. Okay, so level of this enzyme is increased within 48 to 72 hours after the heart attack, right? That is after the appearance of symptoms of heart attack, the level is increased within 48 to 24 hours, 48 to 120 hours, right? Next one is glycogen phosphorylase. So it acts on glycogen. You know what is glycogen? It is a polysaccharide which is present in animal. In plants, it is starch, right? Polysaccharide present in plant is starch. So instead of starch in animal and also in human being, we have glycogen as a polysaccharide. Okay, so normal level is 2 to 62 nanogram per ml, right? So in muscle disorder, the level of this glycogen phosphorylase is increased. And next, coming to enzyme used as analytical reagent. Okay, that is for analytical reagent means to analyze the compounds, right? So especially for the measurement of drugs and also some other substances, toxic substances, these enzymes can be used, right? So it is specific for only one substrate. What are the enzymes used in analytical reagent? Namely glucose oxidase, It is used for assay of glucose peroxidase and also used for glucose assay. Lipase used for triglycerol assay. Cholesterol oxidase used to analyze or assay the cholesterol. Uricase for uric acid assay. Bilirubin oxidase for bilirubin estimation. Urease for assay of urea. The increased amount of urea indicates the kidney disorder, kidney damage, and also bone disorder. Next slide. Next one. Enzyme used as a therapeutic agent, that is for treatment, that is for a transfusion of fresh blood. These enzymes can be used, and digestive enzymes can be used for digestive disorder, and also we can use streptokinase to release clot in the brain. Streptokinase, one of the costliest enzymes used to remove the blood clot in the brain. And also many enzymes are used in cancer treatment, like uricase, urease, these enzymes, right? Namely some enzymes, right? Some examples: asparginase used in leukemia, it is for blood cancer. Collagenase, it is used in skin ulcer, and glutaminase used in again leukemia. Lysozyme used as antibiotic. Lactamase used in penicillin allergy. This ribonuclease used as antiviral agent, right? And one important enzyme, uricase, used in gout, that is gout disorder. So what is gout is nothing but increased accumulation of uric acid in the bone, mainly in joints, that leads to pain in the joints. That is called as gout, right? So for that we can use this uricase enzyme. Next slide, please. Next one is, hello, organizer. Next slide, please. Hello.
Yeah, yes, sir.
Next slide. It is not moving.
Let me check. Yeah.
Okay.
Yes.
Yes, sir. And specifically, some enzymes are used to detect the disorders or disease in organs like prostate. We can use acid phosphatase, and liver and liver parenchyma, that is for muscle and heart and liver. This alanine aminotransferase is used, and muscle disorder, both skeletal muscle, which is present in other parts of body, and heart muscle, we can use aldolase enzyme, and pancreas amylase can be used. And for any disorders or any block in bile, we can use pyrase nucleotidase and trypsin for pancreas amylase. Yes, next slide. Okay, and so the my presentation on the use of enzyme in diagnosis is over, right? And also the use of enzyme in food industry is also over. And in this contest, I wish to thank the scientist, two scientists, actually. Previous one. Hello. Yes. Dear organizer. Yes. Each and every day we should thank the scientists. I used to tell our students in my college, right? The scientists are like our friends and neighbors, right? So, like our friends and neighbors, each and every day, at least 5 scientists' names should be written with their discovery or invention. Actually, in the notebook, right? Like a diary notebook, right? So, in this, as far as this chapter is concerned, I like to thank the 2 scientists. One is Friedrich Kuhne in the year 1877. Actually, he is a German physiologist. Okay, he born in that is Hamburg in Germany, and he is known for the enzyme. Actually, he only coined the name enzyme. Okay, that is Friedrich Kuhne. My dear students, please keep it in mind and please make a note. Actually, he only coined the name, that is enzyme. Okay, this is German physiologist, right? And one more scientist I like to thank, that is Bachner. Okay, that is he only extracted enzyme from the yeast in the year 1897. Also, actually, he did this experiment in the University of Berlin, again Germany, and also he extracted the enzyme from the yeast and he named the enzyme as Zymase, right? And in the year that is 1907, he also got the Nobel Prize in chemistry. Okay, so dear students and dear participants, please keep in mind these two scientists. Please make a note. Next slide. Yes. Next one. I end my lecture with this quotation. The goal of education is the advancement of knowledge and the dissemination of truth. Okay. So whatever the knowledge you acquire, you should disseminate. Okay. Disseminate means to spread out and convey the knowledge to others. Right. So this is the aim of education or the goal of education. Next slide, please. Next one. Yes. And I end my lecture and my deliberation, that is with famous quote of Einstein. Most people say that it is the intellect which makes the great scientist. It is not so. It is wrong, right? It's character. Okay. So character only makes the person a human being and also a great scientist. So with this quote of Einstein, I like to thank the organizer and also the correspondent, secretary, principal, and HOD sir, that is Prem Kumar sir, and other faculty of pharmaceutical chemistry department and also other department like biomedical engineering department in the Paavai Engineering College. Thank you very much.
Thank you, sir, for taking out time from your busy schedule and enlightening us with the knowledge. Today's webinar was full of knowledge and interesting things. It gave deep insight into the topic and also revealed some interesting facts. I'm pretty sure the precious information that Dr. K. Jayaprakash sir gave us will definitely help us in our studies and future. Now, I request our HOD sir to share few words.
Yeah, so thank you, sir. So it was very nice as expected. It was very exhaustive and it was so knowledgeable presentation from you, sir. Thank you very much. And it has been nicely segregated with beautiful slides. The presentation was also very good, and the way you presented, I mean, I don't have anything to say about it because it was so excellent. Thank you, sir.
Okay, thank you once again. Dear students and participants, please make use of this enzyme industry. So not only in food industry, in many other industries and also in diagnosis, that is for the to detect or to screen the disease. Okay, so it will be useful. I think I hope this presentation will be very useful for the nursing students and biomedical and also for the pharmaceutical engineering students when you take up the new project in enzymes, right? Once again, I thank you all.
Definitely, so that was knowledge. I hope that it will give lots of importance for the students when they do their project and other things. Yes, sir, definitely.
Yes, sir. Shall we wind up, sir?
Yes, a vote of thanks will be there.
Please. Okay.
Thank you, sir. Now, participants, it's your turn to ask queries. Make sure to enrich your knowledge by asking questions.
Yes, with pleasure.
Sir, there are questions posted in the chat box. Shall I read out?
Yes, 2 or 3 questions you can read out.
Why do chitosan beads dissolve when you introduce the enzyme for absorption?
This chitin actually it removes mainly many polysaccharide in the especially in the seafood items, right? So because of that, this chitin is used and also it is commercially available. This not only for this food industry, it's also used this chitin and chitosanic compound is also used in cancer treatment also, right? So dear students, when you take up the projects, you can take some of the innovative ideas like enzymes used in cancer treatment and also enzyme which can be extracted as you are from pharmaceutical chemistry, you can extract new enzyme from the medicinal plants and some of the fungus or some of the seaweeds or the blue-green algae, and find out what is the use of that particular enzymes in a new dimension or with novel ideas. You can present your project, right? So chitosan not only for solubility and also for digestion, it is also used in cancer treatment also. Many research are going on in this use of chitin, right, in many cancer studies. Okay. Next question. Yes, please.
Any role of enzymes in natural and induced postmortem biochemical changes affecting meat quality?
Yes, of course. It's a very good question. Actually, after in any type of meat, in order to prevent the oxidation and also spoilage of food or meat items, we can use those two enzymes like peroxidase and catalase, right? So this peroxidase and catalase, these enzymes are excellent enzymes used to prevent oxidation in any type of meat or also to prevent the spoilage of food. Okay, these two enzymes can be used. It is hydroperoxidase and catalase. Yes, please.
It is possible to use this enzyme for stem cell therapy.
Actually, stem cell therapy is actually a different thing. In stem cell therapy, actually, only stem cells like adult stem cells and germ cell stem cells can be used. But in that stem cell therapy, enzymes cannot be used, right? And in stem cells, for breaking down the compounds or to add up the molecule or to bring the molecule in the correct format for the treatment, we can use the enzyme that is one enzyme that is protease, right? So protease can be used in stem cell therapy. Other enzymes they cannot be used in stem cell therapy because we have two types of therapy. It is adult stem cells and also germ cell therapy. In that, only this protease can be used. Yes, please.
Thank you, sir, for clarifying all the doubts. Now
Okay.
I call upon Sneha from final year Pharmaceutical Technology to give vote of thanks.
The best kind of giving is Thanksgiving. I would like to thank all the esteemed delegates of the webinar for your contribution and making this webinar a great success. First of all, I would like to thank our guest webinar speaker, Dr. K. Jayaprakash sir, for making this session very informative. A special thanks to Mr. Prem Kumar sir, Principal of Paavai Engineering College, for providing immense support to make the webinar successful. I also wish to express my gratitude to Dr. R. Praveen Kumar sir, the head of Pharmaceutical Engineering Department, for always encouraging us to achieve our goals with his positive vibes. I feel an immense pleasure to thank Dr. G. Srinivasan sir, the head of Chemical Engineering Department, for making this webinar special by his presence. I deeply feel glad to show our gratitude to our beloved faculty members for motivating us to organize this webinar in a great manner. I must thank the organizing team, volunteers for working hard for the past few days to make this webinar successful. Finally, I thank all the cheerful participants for paying attention to this webinar. Thank you everyone once again for making it a great success. Thank you.
Yes. Thank you very much. And organizers and actually, shall I wind up, sir? Yes.
Yes, sir. Thank you for
Yes.
sir.
Thank you
For
very
your
much.
presentation, sir.
Thank you very much. Be safe.
Yeah, thank you.
Bye bye. Thank you.